I want to welcome everyone today to Sona Nanotech's investor webinar. I'm David Regan, Chief Executive of Sona Nanotech, and I'm delighted to have with me today our Chief Scientific Officer, Len Pagliaro, and our Chief Medical Officer, Dr. Carmen Giacomantonio. Some of you may have met both before in previous webinars. Now we're a team all on the same page, having Carmen join the team about a month ago. Where I want to start with this is that a year ago, we talked about building a mountain of data in order to progress this cancer therapy that Sona has. With each study, we're effectively de-risking our path forward. And of that path forward, efficacy is, we believe, our biggest challenge, proving that this therapy can work in humans. And we now have a great deal of the evidence that we need to give us the comfort to plow forward with a very aggressive plan to get it into humans. So we're incredibly excited about that and really keen to dig into some of the data with you today in a very detailed way. Of course, the therapy that Sona's developed proposes to take the fight to cancer on a nano level with an elegant treatment that aims to eliminate the cancer without cutting, radiating, or poisoning the patient, and very much activating the immune system in order to leverage that and have the body help fight the cancer itself. Our therapy involves the administration of gold nanorods directly into tumors, an application of near-infrared light that creates a heat inside the tumor, killing the cancer cells selectively and activating the immune system through a presentation of new antigens. But we'll get into more detail on that right now. Flicking forward, we seek safe harbor and with these forward-looking statements. And with that, I'm going to kick it over to Dr. Giacomantonio, who has had a very busy week presenting some of this data to an industry-leading conference on immunotherapies, on the developing immunotherapies, and perhaps you can speak to that as well, Carmen. Just a reminder to folks, there is a chat button here at the top of your screen. There should be. Feel free to put questions in, and we'll get to them as we can and as appropriate, but certainly we'll dig into them at the end as well.
So, hi, everybody, and thanks, David, for that. So what I'm showing you here, I thought the first couple of slides worth showing is the value and efficacy of intralesional therapy alone. And so this first patient is a patient with melanoma, is back all the way to 2011, who had had a number of therapies fail her and ended up with this kind of widespread distribution of what we refer to as in-transit melanoma, with some big raised tumors and some smaller specklings of tumors. And at the time, we had interleukin-2 alone, and we treated, we knew about the relationship with the immune system and melanoma potentially, and we played our hunch and treated this by injecting those lesions. You can't inject every lesion, as you can imagine. There are hundreds of lesions there, but you can inject a grid-like pattern throughout the leg. And over the course of several months, we were able to clear the disease, and not only clear it, but keep it clear. And she's remained disease-free throughout her life. So this is the first indication, the first kind of testament to the fact that intralesional therapy alone can cover more than a single lesion, and was motivation for us in my clinic to continue trying these innovations. Next slide. So moving on to, branching into new horizons, many of the patients I see, not so different from the person I just showed you, but have significant comorbidities. In other words, patients with diseases like this one, that is actually a squamous cell cancer. Why show you a squamous cell cancer? To show you that immune therapy, the type of the cancer isn't nearly as important as the context of the cancer. And so this person who couldn't have surgery, she's on oxygen, as you can see. She wouldn't tolerate toxic immunotherapy or even toxic chemotherapy. And so we chose a local approach with her. We injected this lesion with IL-2, but we added a topical. So we added a known innate immune activator, imiquimod. It's a toll activator, and to simplify the matter, the toll-driven system is your innate system, the one you're all born with. and that would see patterns of danger like a cancer cell, like a dying cell, and respond to it. And so the combination of the danger responding innate system and the interleukin-2 driving the immunity whole part and parcel from left to right, eventually cure this person of her disease. And to this day, I still see her and get pictures from her daughter, and she's disease-free. Next slide. These are the cases, though, that so we're always excited when we can bring elegant solutions to cancer, particularly melanoma. This gentleman, also a patient of mine, unfortunately had the whole top and second shelf of what we have for melanoma. He had ipilimumab, which is, of course, the checkpoint inhibitors we hear about all the time, nivolumab, another checkpoint inhibitor that we hear about, failed that and progressed to the left. The picture to the left is the first time I saw him. You can see from that picture that he has a small tumor on his cheek, and the gland in front of his ear is starting to get bulky. We started him on interleukin-2 and BCG, yet another innate activator. BCG is a tuberculosis vaccine that we've had some very good success with melanoma. And despite the combination of literally everything that we have to offer, his tumor progressed and he succumbed to his disease. And this is really the motivation. This is the challenge that we have to overcome. And this was our strategy pretty much from the start with the gold nanoparticle that Sona has and what it potentially does for patients like this. Next slide. I'm going to ask Len. Len is our Chief Scientific Officer, and this is actually his backyard. This is his area of expertise, and he would tell us about what this little nanorod does.
Thanks very much, Carmen. So the gold nanorods they're wonderful little things. They're extremely little. They're truly nano. You can't see them in a light microscope. You need a light microscope or an electron microscope. And what they do is they convert infrared light into heat. They basically act like little tuning forks. They absorb light and they re-emit it at a different wavelength. And so if they're tuned the right way, and Sona's nanorods are, then they re-emit infrared light as heat. And you can concentrate a lot of heat in tissues if the nanorods are in the tissue. One of the big characteristics of the Sona nanorods is they're manufactured without using toxins that are used in other manufacturing processes. And so they're extremely biocompatible, they're safe, they're biocompatible, and they've been highly vetted by the Nanotechnology Characterization Laboratory, the NCL, through a series of tests for physical chemical testing and cytotoxicity and hematology and so forth. Extremely safe material for injection into the body. Now, targeted hyperthermia is our treatment, and it combines two components. It's a two-component medical device system. The first is the nanorods, which we just introduced, and those nanorods are injected into tumors intratumorally. They're metallic gold, they're polymer coated, and you can't see these in a light microscope. So on the left is an electron micrograph of a red blood cell, which is one of the smallest cells in the body. And those little tiny specks inside the red blood cell are the nanorods. On the right is a schematic of how this would work in a patient. The nanorods are injected into the tumors directly, and these are for superficial tumors such as these melanoma tumors. And then that region is illuminated with infrared light, which comes from a laser device that we've developed. Now, the combination of the infrared light and the nanorods stimulates heat, release of heat inside the tumor. And it turns what's called a cold tumor into a hot tumor. And it really does this in two ways. One is thermally, temperature-wise, you increase the temperature of the tumor, but you also activate the immune system in that cell. You activate antigen presentation, you activate T cells, and this now becomes an immunologically activated tumor. An important aspect of this is that it's done at a moderate temperature. And when we say moderate, we mean between about 41 and 48 degrees Celsius, the target being 44, 45 Celsius. And so this is hyperthermia as opposed to ablation, very high temperatures. This is where you get this activation of the immune system.
So, of course, we've gone into this study through Dalhousie University with Dr. Giacomo Antonio leading as principal investigator. And as indicated, we've been seeking to build a mountain of data to support our applications to regulators. We've been patient and we've been rewarded for our patience. And Len, perhaps you want to go through kind of the summary of what we've established so far.
Sure. So we've known for a number of years that targeted hyperthermia can shrink tumors and increase survival. And that, of course, is a very good thing. What we really know now through the studies that Dr. Giacomo Antonio's lab has been doing is several very important things. First of all, we have direct evidence for stimulation of the innate immune system. That's extremely important because you're using the patient's own immune system to fight the cancer that they have right now. The second thing is we now know, again, based on the new data, that THT upregulates gene expression of innate immunity and inflammatory response pathways. And this, again, is significant because it shows the mechanism behind the tumor shrinkage. And then the third thing that we've seen is that the combination of THT and immunotherapeutic drugs can result in an abscopal effect. And that means the reduction of tumors that haven't been treated directly but are distant from the primary tumor that's been treated, those shrink also. And so these are all very, very important things, both in terms of cancer treatment, but also mechanistically, and they'll be very valuable when we go back to the FDA to get clearance for initiating clinical studies.
So these are, some extraordinary things that have come out of this study, and so we're going to dig into each of these areas one by one to detail them a little bit further for you, focusing in on the implications as to what some of the insights really mean for the company going forwards. Carmen, can I call on you to come to take up the gauntlet here and talk us through what we're seeing?
Yeah, so essentially, and I apologize for the most models, but this is the workhorse in the industry of immunotherapy across the board. And I'm just going to give a little background. The reason we use mice, and mice aren't people for sure, but their immune systems, we're highly conserved across the species. And so the principles of immune activation and immune response are conserved in the mouse, similar to that of the human. And of course, the mouse allows us to do in a humane way the research, ask the questions we can in large numbers, so that we're proving with each batch of experiments the same results are being seen over and over and over, confirming that this is not just phenomenal; it's actually predictable. And so what we see here on your left, you see when we grow these tumors in the mice, we grow a tumor that's implanted into the mouse under the skin, and then we let it grow to a certain point where we know we can actually we can inject the tumor, we can heat the tumor, and then on the right side, we take one of the two. So there's two tumors here on either flank. We take one of the tumors and we treat it. Now, the reason we only treat one is because, well, a number of reasons. One is we want to see what the direct effect of the treatment is within the tumor. And two, we want to see, does that response within the treated tumor translate to something that's systemic? Does that response lead to a systemic outpouring of something, hopefully immunity, that sees the other tumors? What we see here on the graph to the right is these are tumors that are treated with they're impregnated with gold nanorod, and then they're treated. And then what you see is, quite astonishingly, that 24 hours after a single treatment with the hyperthermic therapy, that this tumor, and then what you see on the left in the treated tumor is the tumor completely flattens. This is essentially there's still tumor volume here. What we're measuring, though, that's a scar. And so when we see tumor response, we measure it in 3 dimensions: height, width, and length. And then there's a formula that helps us to calculate, to standardize everything, to give us a tumor volume. At the end of the day, when we treat, the tumor essentially disappears or goes down to a cellular level. And what we're measuring is mostly scar tissue and residual matrix that the tumor would have had grown into.
Okay.
Go ahead, next slide. So so in this slide, what we're showing is that we see, so what happens over the long haul? The question you should ask is, so we give this one treatment, is it all gone, and is it all gone for good? So when you follow this experiment we did, we took tumors, and this is a B16 melanoma model. B16 is the industry standard for treating melanoma. It's a very challenging model to work with because it actually doesn't generally drive a good immune response. And so that makes the challenge for us better because we're trying to drive immunity where it doesn't necessarily happen. If you see this graph, you see the gray is the control where this is just gold nanorods, no treatment. If you see the gold is gold nanorods and then lasered with a single laser. And then there is the gold nanorods with a double laser. Now, with a single laser, you see that there's good tumor responses. If you look near compared to the controls, this laser has had a brilliant effect with activating the gold and causing tumors to shrink or fail to grow, if you will. But over time, there's escape, and the escape is because there is we know now we're driving an immune response, but you need to continue to cajole that immune response. You need to do something more. So we asked the question, what if we just added a second treatment with the laser? Can we activate residual gold nanoparticles, and we activate residual cell death? And so we did that. We put a second laser treatment only, no additional immunotherapy, and for sure we completely flattened the tumor. What's again seen at the end of the day in that graph at 20 days is that's essentially scar tissue. There's no viable tumor. So this was to us very encouraging in two things. One is we give a single gold treatment. The gold particle hangs around in the tumor or in the tissue long enough so that we could take advantage of its presence and drive second immunity, second energy into the tumor. Next slide. Of course, then back to the first question, we give a single gold treatment and activate it with the laser. What is the effect over the long haul? Well, you can see from this graph there. This is again the B16 model. If you look in the blue, that's controls. That's just control means we've put the tumor and we treat it with just saline, nothing of any immune consequence. The second line is interleukin-2. And when I said to you earlier that this tumor is not immunogenic, interleukin-2 is an immune cell activator. It's one of the first immune therapies ever developed back to 1985. We've been using it in the clinics for all this time. It's the mainstay of my treatment in the clinic. And like the gentleman that you saw, he received interleukin-2, and it didn't have a huge effect in and of itself. And that can be very frustrating in melanoma. The third line down in that is these are the tumors that we treated with gold only. I've already shown you that this is effective. So the gold therapy did as we expect. It had a significant impact on tumor growth and volume. The fourth line is kind of the exciting line, if you will. And it's that, so as I said, the gold will activate an immune response, but the immune response has to be enabled. It has to be driven over time to go to its complete and total effect. And so the green line is what happens when you do that. And that's gold laser, and then sequential treatments with an immunotherapy that's already on our shelves today, showing that when we do it in the right way, when we activate the gold nanoparticles and we've activated immunity in the tumor, we can continue to drive that immunity within the tumor and annihilate or close to annihilate the tumors. And that's what we saw in this B16 model. It's very similar that you might remember what we showed you in the 4T1 model, where this immune response is the combination of gold nanoparticle driven energy, hyperthermic energy, immune activation, and then immune enablement with the interleukin-2. Next slide. So what is this gold doing? So the first question, and we got many questions since we presented to say, well, what's the gold doing? Is it just heat? Is it in and of itself an immune agonist, an immune activator? And the answer is yes. And so what we did was we took the 4T1 model and the B16 model, and we're looking at PCR. We're looking at RNA to see in these models what genes are being turned on as a consequence of energy driven by the hyperthermia from the gold nanorod. So if you look at the 14-1 model in the controls, it's essentially that bluish, purplish coloration saying everything is quiet. And that panel on your left is the panel of genes that are typically associated with an innate immune response. In other words, if those genes are being turned on and being upregulated, your innate system is busy doing something. In the 14-1s, we activate with the gold nanorod hyperthermia, and we see that all of those genes are being turned on to one extent or another. If you look, the more yellow, the more gene is being activated. And if you look to the left and you know anything about the biology, these are the genes that are typically confirmatory of an innate response. Similarly, in the 14-1s, we saw a robust immune response across all of those genes. And this is the evidence that this gold nanoparticle in and of itself is an immune activator. And that to us has been a revelation. And I can share with you, we just presented at a meeting, it was to everybody working in this field, that this is in some ways the missing link in that it not only activates the immune system, but it's doing what we wanted it to do, and that is exposing new antigen for that activated system to respond to. Next slide. So where's the proof? And the proof of the pudding is in the taste, as they say. So we looked into the tumors to see, did we actually drive innate cells into that tumor just with this? So this is just GNR-induced hyperthermia, no additional immune driver. And these are dendritic cells. So dendritic cells, what are they? These are the antigen-presenting cells. So the cells, if you have a cut on your hand, if you have an infection, if you have a tumor in a mouse with gold nanoparticles and heat, and you injure that thing, the cells that are dying have to be cleaned up by the innate system. So these dendritic cells, as they eat up, the other name for them are phagocytes, as they eat up the debris, they present that debris to the immune system to generate that response that we all want. In a benign situation, it would be a cut, and it will keep on going until the cut is healed. The same thing with macrophage. They're also, macrophage are the other phagocytic cells. They chew up debris, they control, and macrophage are kind of the cells that are responsible for maintaining harmony within the tissue, that whatever tissue it is. And so we drive macrophage into the system. And these are, when you look at this over PBS, that's phosphate buffered saline. We injected the tumor with phosphate buffer versus gold. There's far more dendritic cells and macrophage within the tumor. Next is the next cell we looked for was the M1. Now, what's an M1? That's a memory cell. So we don't typically think of an innate immune response as something that generates memory, but we know that memory is a part of an initial response. And so what we're showing here is that the M1s, the M1 macrophage are the cells that are being driven to, this is kind of like angry tumor cells. And these are the angry tumor cells that will drive you further down the road to a more lasting response. So M1 means tumoricidal. If we saw lots of M2s, we'd be concerned we didn't see the M2s. So we have a strong M1 macrophage response. And then the final slide is this is the kicker. What everybody wants to see in an innate response are these natural killer cells that come in and chew up these tumors. And we saw what the bar over the top is showing, that is statistically significantly better than control alone. And so we've shown through the gene activation profile, and we've shown through the cells that are actually infiltrating the tumors with gold alone, that we are generating an innate immune response with the appropriate cells infiltrating that tumor to essentially do what we hope it does, and that is tear the tumor down. Next slide.
So, Carmen, if I can just ask one more question, I think you're just getting at that in the last comment. But we previously had the volume data showing that tumors shrunk.
Right.
Why is it important to then we had to wait some time for the biomarker data to delve into what's going on? What difference does it make? We knew already that the tumors were shrinking.
Well, yes. So if the tumors are shrinking just because even if it's just because we're activating a cell death cycle, the problem with that is it has to translate. It has to translate to something that generates immunity that will be there for residual tumor or recurrent tumor growth. And so if we can put gold right now, we could go into a person that has a solitary tumor with no chance of metastasis. This would be a great idea for a sideline discussion. And we could treat it with these and probably shrink the tumor down elegantly. So if we want to shrink it down to make a less traumatic surgical operation, this would be a good application. But what we're really looking for here is to treat a cancer that has a metastatic potential so that we know we're treating the source of the disease, and we're also treating the potential cells that are going off into here and there. And so these cells are telling us we're generating immunity. They're the complementary components of that tumor clearance process.
So that kind of sets us up for this next slide, which is a little bit of the ta-da moment. If you could take us through.
So this is it. So this, and we were, just like I was super excited. Of course, I'm super excited, a lot of stuff in immunity, but when we did the 14 ones, we saw we flattened that curve. Well, this is so we've done a kind of an elegant experiment here to show that we're generating a systemic response. That's that abscopal response that you hear about all the time. Something abscopal literally meaning you're seeing a response beyond the site that you actually treated. So in this model, this is again the B16 melanoma model. We have the controls where we just used PBS. We had a tumor, a primary tumor, and then we initiated a second tumor shortly after starting treating the primary tumor to show A, that we have systemic immunity, and B, we can control regrowth or ingrowth of new tumors. In the control, we had no effect. Interestingly, and somewhat frustrating from a science perspective, we have no effect with IL-2 alone. Again, we're speaking to the fact that B16 cell is cold. And cold meaning the immune system doesn't see it like a good vaccine, like a good antigen. It's just doesn't generate that interest to drive the immune system all by itself. We added the gold, and we saw, you can see it moves the curve to the right between the 15 and 20 points there. But there's regrowth because, again, we need something more to drive that response. This is systemic response. We're looking at the other flank, not the primary treatment, but the other flank where we started the second tumor. And then if you look at the ta-da for sure, is that the green one, in the mice that we had treated with IL-2 and the gold nanorod hyperthermia, we could not generate new tumors. So we've essentially not only generated this abscopal effect, abscopal meaning we've generated a systemic response. This is the vaccine effect that you hope you see with immunotherapy, meaning that not only can you clear the tumor, but other tumor cells that may end up in circulation aren't able to engraft. They aren't able to establish a home and create a new tumor. So this
suggests that if there were tumors that had not been yet identified, that if we treated one tumor that was identified, others would be impacted by this,
right?
Even if we didn't know they existed yet.
Right, exactly. So that's the whole point. This is the whole point of clonal distribution in cancer. These give off all kinds of daughter cells, and the daughter cells may or may not be within the pocket where it all started. They can end up in circulation. So this is a systemic response. These tumors are not growing because there are circulating cells preventing them from growing.
Excellent.
So in summary, I'm introducing a new word here. This is STING. STING means these are stimulators of interferon genes. Interferon is the kind of the linchpin of inflammation and immunity. So you need that combination. The hyperthermia from the gold nanorod induced the STING innate immune pathway. I showed you that with those genes being activated, and it reduced the tumor volume dramatically. And it wasn't simply because the tumor was dying from the heat trauma. The combination with the IL-2 not only brought that 1-2 punch so that we actually had tumor clearance, we had the sufficient immune response to flatten and level the tumors, but it created a durable response where we couldn't initiate new tumors in the contralateral flank or otherwise systemically elsewhere in the mouse. The other thing is we didn't show you the data because there's tons of data to show you, but this was all at the end of the one when we introduced the interleukin-2 and the gold nanorod. This is all CD8 toxic cell death. CD8s are the T cells of your immune system that fight cancers. And essentially what that did was those CD8 T cells are responsible for the ultimate demise of the tumor. The other thing is, and I put this in, this is here, this PD-1 expression. So why is that important? PD-1 is an indicator of an activated, engaged immune response. So PD-1 stands for Programmed Death Receptor 1 expression. And it's a death receptor because once a T cell has been turned on for 24 hours, it exposes this receptor to say, I should stop working, but if the antigen is still there driving me, more cells will come behind. Do you know what I mean? So it's a checkpoint. In an immune engaged in a relationship with a cancer, you don't want that checkpoint to be working, so you can block it. So the immune response that we generate with the gold nanoparticles and IL-2 generates an immune response that we can even augment further with the addition of a PD-1 inhibitor. And we have research underway now to prove that that's the truth. And so what it opens up is an opportunity for the patients, like my first patient that failed PD-1 therapy, to maybe benefit from PD-1 therapy in combination with the new antigens that our gold nanoparticles are potentially presenting to the system. So this is over the top exciting from a cancer solution point of view. So, and again, the abscopal effect that we've been seeing is testament to the fact that this is not only a local therapy, this is a systemic therapy enabled by a local process.
So, Carmen, obviously extraordinary, and particularly that chart show the flattening of the line for a tumor that is absolutely untouched, untreated, and yet will not grow, whereas cancer, as we know, just grows so rapidly. That is extraordinary. What has to happen for this to get attention? When does this important? I mean, obviously it's early days. We've got 2 models. This is mice.
I think what happens now is, so we're repeating some of the experiments so that we have that robust, statistically significant data. And what I've shown you is all of that. And when it's past scientific rigor, we're submitting this in the next couple of weeks to a scientific journal, Frontiers in Immunology. It's a journal that it's a very highly respected journal. It's got a solid impact factor, and it's kind of a world platform journal that people that look at to see what's on the horizon. And we'll get this published in that journal, and that will give us the peer review that we need to generate the bigger interest in terms of the rest of the world knowing what we're doing. The other thing, though, is I will tell you, we just presented at the STING meeting. The meeting focused just on this, and we were in San Diego last weekend, presented, and we featured beautifully with all of the presenters. In fact, we generated enough interest to warrant collaborations that we're now working with companies in China, another startup in the States that are both in the clinic. So this is a solution for many people looking at this problem from many different perspectives. There is no one solution to cancer, but there are a number of facets of immunity that you can exploit to control cancers, and this is an enabler to many pathways. So but the scientific and peer-reviewed publications are validating in that it takes it out of our hands to be the judge.
Right. So it's got to be in the study. It's got to be replicated. And it's not just a phenomenon, and then peer-reviewed once submitted to a journal. And presumably that's when folks get interested, when it's actually been picked up by a credible academic journal.
Yeah.
Again, what's the importance of having shown this in not just triple-negative breast cancer, as we reported on a couple of weeks ago, but now with melanoma, second cancer model?
So this is this term I teach the residents and tell the patients. This is tumor agnostic. It doesn't. There are many treatments on. We know now that if a person has lung cancer, they get lung cancer treatment. Person has ovarian cancer, they get ovarian cancer treatment. This has nothing to do with the phenotype of the cancer, if you will, of what the cancer looks like. This has to do. It exploits a principle, and that principle is this. Every cancer produces cells that make abnormal, dysfunctional protein. And that dysfunctional protein is essentially an antigen foreign to your immune system. And if you can show your immune system that novel, foreign, dysfunctional antigen, you can create a response to the cell that's carrying it. And that's what cancer immunity is. And so that's what we have. This is agnostic. We know we can find those antigens in any cell. We just have to be able to get into the tumor. That's why we're giving intralesional injection. We're taking these tumors and we're going right to the heart of it. If you read anything in the journals about challenge, it's the tumor microenvironment. And the best way to overcome the challenges of a tumor microenvironment are to directly annihilate it. And so this is the big news.
Excellent. Okay. A few other questions here, including, what makes this novel, and what is the closest thing out there in the industry, in science, and in the clinic that effectuates a commitment of antigens?
That's a great. So what makes this novel? Two things. One is it's hyperthermia, not ablative therapy. So most of the things that are in the clinics right now to treat tumors require some kind of ablation. The first group that ever showed that you can generate an immune response through ablation are the radiation oncologists. They radiate tumors, the tumors die, and the dead stuff gets presented, and lo and behold, every now and then you get an abscopal response. And we used to say, kind of tongue-in-cheek at the meetings, they were the unicorn responses. But if you don't ablate the tumor, if you actually tear down the tumor, and the kind of death that happens with hyperthermia is, it's a programmed cell death. So the cell is actually dismantled by surrounding cells and itself. And so the protein is intact enough so that the immune system can see it. And when it sees that abnormal protein, it has a response. And if you can, so if you know, this is the special sauce, and what we immediately took advantage of. If you, at that point, at the right point in time, activate the immune, overdrive the immune system with a something, a IL-2, a PD-1 inhibitor, another toll activator, blah, blah. There's another, again, this is then you have dramatic immunity. So it's unique in that we are exposing novel antigen in a completely new way. And there's really nothing in the clinics today that's doing this. This is, now, are other people going to want to do this? Absolutely. And there are some other ways, and we could talk about that, at a journal club about what are the alternatives. But we are a long way down the runway now with evidence of how this works. We have mechanism of action. We can show it over and over. In fact, my lab is doing a more detailed dive into what actually the science is of hyperthermia. Hyperthermia has been around a very long time. I've been doing hyperthermic isolated limb perfusions for melanoma since 1997. So, and we've seen tumors shrink, but they don't shrink for long. They come back because it's an incomplete death in and of itself. If we knew how to drive immunity, that would have been a good way to do it. So there are other ways to do hyperthermia, but the way we do it in the operating room is to tie off your leg and then isolate it and then heat it up. And so it's very toxic, very brutal. This is elegant. It's tumor specific. The other thing I do is I use hyperthermia in abdomens for cancer. And again, we have to heat up a whole person to get to this effect. This takes the horizon for this kind of thinking is as big as your imagination. So, yeah.
Len, a question for you here, because there's a question as to what are the critical elements that makes this Sona's therapy, and how is it protected?
How is it protected from an intellectual property standpoint?
I think so. I think the sense of what stops somebody else from doing it too.
Yeah, no, so that's important. Well, I'd say a couple things. First of all is the nanorods. We've known for many years, well over a decade, that nanorods are very good transducers of infrared light, turn them into heat. But getting nanorods that are consistent, that are uniform in size and shape, that have a consistent polymer coating, that are sterile, non-toxic, and so forth, free of endotoxins, that's a whole different ballgame. And that's what Sona can do. Sona's manufacturing process enables manufacturers, real manufacturing of nanorods, and scalable manufacture, which is the next step that we're working on now, is to get that up to a much larger scale. So that's an important part. There are patents around the nanorods themselves. And then the second part is the treatment, putting it all together. So for the treatment, you need the light device, and we've got patents underway around the light device, and then also around the treatment protocol itself. So all these things are important to bring it together. One part alone won't do it. And that's I think it's one of the things that Carmen mentioned. It's an elegant approach because that it requires several components to work together.
Right. And we, of course, have developed a great deal of expertise in terms of how to deploy this. And that's part of the provisional patents that we've been filing along the way here in order to protect ourselves. Another question, and looking forward, is to an eventual treatment, and whether there'd be multiple infrared light applications, and whether this would be combined with another immunotherapeutic drug of some sort. Throw that out to either of you.
Yeah, so that's a great question, and the answer is yes and yes. So, what we're envisioning is this. So this is small tumors, 2-3 centimeters. You can treat with one treatment adequately. Large tumors are probably going to need more than one treatment just because of the sheer volume. And we got maintaining heat across the tumor is there's a skill to that you have to apply. So the safest way to do it would be to treat multiple treatments, 2 or 3, and we don't know yet. But this is, as I said, definitely will be combined with other therapy like intralesional IL-2. And I'll give you a great example. Right now at Dalhousie, we have the first intralesional IL-2 treatment protocol for triple-negative breast cancer in North America. And we're seeing some response. And this will be a natural runway for the follow-up study to show that we can enhance the annihilation of those early primary tumors that don't respond to IL-2 alone, just like we're seeing in our models, to the combination. So we're super excited about that, because that's going to be, so that's one combination. But the other thing, as I said, is these immune responses generate PD-1 expressing T cells. So the natural and literally every immune therapy today is looking for combination with one of the checkpoint inhibitors, namely the pembrolizumabs that you've all heard about in the world, or the nivolumabs, the PD-1 inhibitors. So this is a clear and obvious complement to that strategy, hopefully enabling a new immune response that would increase the response rate overall for the patients. And so definitely. And with the publication, and we actually, full disclosure, we actually have some conversations beginning to happen with these companies now at one level or another. But with the peer-reviewed publication will come the legitimate commitment or interest in these kind of clinical collaborations.
Excellent. So a question here also on what we see as our biggest risk or biggest hurdle going forward to getting an approval from regulators to go into humans, into the clinic, as we say. And I'll address that to some extent. I mentioned it off the top of the call. Of course, we saw initially the biggest hurdle was the efficacy, being able to show efficacy, strong data on the efficacy of the therapy in multiple types of cancers. We have that, as we've indicated in the past. We're going into colorectal cancer next, but based on what we have, we're not waiting for that data to plow forward with the set of studies that are required by regulators to enable us to go into clinic. We're not seeing big hurdles going forward. Much of it's all safety related, biocompatibility related, and while there are risks there, we saw the biggest hurdles as the efficacy. Going forward, Len has already done studies in the past on clearance, and so chronic toxicity is something that is very important from whether it's Health Canada or the FDA's perspective. And Len, what is our chronic toxicity history shown in the past?
Well, we haven't done a formal chronic tox study. However, what we do know, and this comes from data from the NCL studies that we've done, is that the material is cleared. The nanorods are cleared in both the urine and the feces in a rat model, which is a pretty good clearance model, pretty good fidelity with humans. The clearance is much slower than accumulation. Of course, accumulation is very fast. It's an injection. But the fact that the material is cleared is very promising in terms of chronic tox, because it indicates the material will very likely not be in the body for that long. And so if you're worried about 12-month chronic tox, well, if the material clears in a month or two, that's not a big deal.
Right. And certainly it's inert in any event. And as we know, we've got, we've been putting gold in teeth for years. So there's lots of history associated with that. Yeah, there was another question on expulsion of gold nanorods. Much has been made about potential abscopal effect. What kind of abscopal effect has the process noted? Why is the antigen presentment important? What is the potential of exposing this antigen in this Sona therapy? I think we've covered that to a large effect. But do you want to say anything else, Carmen, about the abscopal effect? And
I think that's really the key. That's one of the big motivating drivers of all this research is that we're seeing the abscopal effect is essentially the systemic effect. It's we're seeing that we're generating systemic immunity because we're exposing new antigen, and there's absolutely no downside to that. That is the essence. It's the missing ingredient in all immunity is that strong antigen. And so, yeah, this is again, I've said it too many times now. This is a very elegant way of generating or exposing new antigen. And I'll speak to the other question about runway and hurdles to get into clinical trials. Our pivot to inject directly into tumors has really cleaned up the systemic risk question to a large degree. Most of what we're injecting is it's high volume, high dose, relatively speaking, into a tumor, and minuscule, if any, measurable dose into the systemic circulation. And so we're very confident with this model, and so was the FDA gave us some indication that this is a much more palatable first in humans strategy. And in the meantime, we're doing all the systemic. Len has been busy having all the systemic toxicity work done so that we can move into that as we go onto other horizons that we'll be happy to share with you in coming months. But yeah.
Okay. And another one here. Looking back in the fullness of time, what do we expect that will be looked at as being groundbreaking in what it is we're doing?
Antigen, new antigen presentation. This is definitely groundbreaking. We're showing that this is what's going to distinguish this from everything else that's happening. We are generating novel antigen, and so some of the work you'll see coming down the pipe is the elegant research to show that the T cells, the cells that are killing the tumors, that we generate through activation with the gold nanoparticle hyperthermia plus immunity is a different T cell than the T cell that would have been stimulated through some natural process if we just put the tumor into the animal. So that kind of research will not only confirm, will show that we can generate new immunity to a tumor over and over. See, the idea is that there are hundreds of millions of odd proteins in these tumors. Once the immune system engages a type or a group of protein, the engagement is over. There's no new immunity happening. Now that playing field is dominated by whatever players are on the field. You can generate new immunity within a tumor by simply starting the process over. And mathematically, there's going to be new cells and new tumors engaged. And if that engagement is more powerful, more stimulating to the immune system than the one that's already in place, that's the one that will dominate. That's how, when you think about every year when we get our vaccination for the flu, we say the chances are we've got the right antigen, but sometimes we don't. So you get your flu vaccine and everybody gets sick. It's not because the vaccine didn't work. It's because we used the wrong antigen. And that's a mathematical thing that the actuaries and the people that are planning these things around the world have to wrestle with. These principles apply to everything that has to do with antigen and immunity.
Good analogy. Okay, well, we'll move forward here. If there are any other questions, please feel free to hit that chat box and ask questions. Folks, I think have seen in previous presentations, we've got a strong team supported by a very capable board, a board that hits really above its weight in some of the experience. The team, we've got access to outside advisors, and that has been what has helped us guide our program to where we are today. But looking forward, we have numerous milestones looking forward. The key thing is we want to get this into the clinic as quickly as possible, and we now know exactly what we need to do. We've had regulatory consultant NAMSA, who's been essential to our process in terms of devising the program going forward. We've consulted with U.S. panel of physicians and payers on what we need to do. We've met with the FDA, we've diligenced partners, and we've selected partners, and we're now ready to proceed. In fact, we have started already this week with the safety studies that will be required by regulators. And we have the goal to get into human studies next year. So again, lots of work to be done, but we have a very strong plan. We've got our hands around it, and there's nothing in it that scares us terribly, particularly after, number one, having the data from these 2 animal studies from Dr. Giacomantonio's laboratory, and secondly, having been at the STING and TLR targeted pathway summit, and seeing what the leading immunotherapies are being developed in the world, and recognizing and getting the accolades from those in attendance and in the know as to how exciting this is. We know exactly what we have here, and we know exactly what we need to do to go forward. So we look forward to plowing ahead and reporting back on a regular basis. One other question here. Please review again the issue of FDA regulation as device or drug and what those consequences might be. Len, I'll let you take that one.
Yes, we're clearly in the device category with the current material. The important criterion is whether anything in the injectable material enters a metabolic pathway in the body. And with the current material, it's polymer coated, gold nanorods, it's neutral, it's inert, it's non-toxic, and so forth. It doesn't involve or enter into any metabolic pathway in the body. So that is clearly a device. And that's a good thing from several perspectives, not the least of which is the regulatory perspective, because it means that 1 animal model rather than 2 animal models will be sufficient for us to get clearance to go into the clinic to begin clinical studies.
That's right. And we know we've already had guidance from the FDA for one, suggesting that rat studies would be sufficient for us, and we don't need to go into swine, which is extremely helpful. There's a question here in terms of the risk of doing overseas clinical trials versus trials in the US. At this stage, we're looking to get into human as quickly as possible, but we need to do it in appropriate fashion. And we're in tight consultation. We'll be consulting again with the FDA and in due course with Health Canada as to what they're going to need to see to go forward. But if we look at the catalysts, we will have rat studies coming up, and we'll see to the extent to which this translates from mouse to rat, and then through these studies into human. That's what we need to show, and we'll do that as quickly as possible, because each of those items become a significant catalyst. That together with, of course, the publishing of the data and the peer-reviewed nature of this study. Finally, looking forward, the goal here, of course, is to help those folks for whom no other therapy has worked. We're going after arguably one of the hardest areas in terms of late-stage irresectable melanoma, for which nothing else has worked. But at the same time, if we can prove ourselves there, we can work backwards into all of these cancers, or many of these cancers, we believe. This chart shows one study from Merck indicating what is frankly a very average low response rate to the leading immunotherapies out there right now. What we've seen and what these studies hint at is the fact that we make those, at least one of them in the case of interleukin-2 therapy, work much better because we activate the immune system through that presentment of new antigens, enabling those drugs to go in and do their job better, and enabling the body to do its job in terms of going in and fighting it. We think that we can effectively raise the response rate here, and if we can do that, we can change the lives of millions, and that's what we're trying to do here. So with that, unless there are any other questions, we want to thank you for your time and welcome any questions anytime. We'll look forward to keeping you up to date as we proceed with not only our safety studies, but also a continuation of other studies. We've said this is a platform technology. Right now, we have targeted hyperthermia, but we see, and I think perhaps you can see from the efficacy of it, the potential applications are many-fold, particularly in the world of antibody drug conjugations. We get a lot of questions about that and what we might be able to play with. Carmen alluded to the fact that we've had inbound interest from multiple parties who see the benefits that we can bring, and we're going to be looking very selectively as to who we might want to work with on that basis, and look forward to reporting back. But I want to thank Carmen and Len, and thank everyone for joining. We are here if you have any questions, and we are excited, but we will have our noses to the grindstone, as has already started this week. Thank you for your time.